Smells funky in here.
Do I need to videotape this?
Excuse me.
All right, y'all, come on over.
Wait, let me find my pen.
Let me find your phone.
Yeah, I'm still yours.
Ooh, it smells great.
Oh, it smells like fruit candy.
It does smell like some candy.
Does it really?
I'm so sorry.
No, it doesn't.
It does.
No, it doesn't. Is it me?
Not with 5 Day Coffee. No.
I don't think so.
Doesn't smell sweet, so I'm just making fun of it.
Maybe it's me.
Yeah, no doubt.
That's in your way.
Yeah.
That's in your way.
Oh, that's a perfect video. Can you send that?
I will.
And you're gonna upload that on YouTube or on
I can do it. It just takes stupid forever to do it on YouTube, but I can do it for sure. I did it on that.
What carrier do you have?
I use. Verizon.
Oh, okay. Oh, Verizon's
You
pretty
don't have
good.
Wi-Fi?
I have horrible Wi-Fi in the country, but even my mother-in-law's it took a while. That's here in the that's here in the city.
They fell off.
But it went through. It kept telling me that it was still trying to upload.
Alrighty then, we had some problems here. Listen up.
To hidden your gettings.
Listen, folks, we had some problems. What's going on?
I can't see him.
Oh, my bad.
No, it's okay.
We had some problems with the plates for some reason. Didn't work all that great.
You gonna move it? I'm gonna move this Sylvie here. Oh, that's okay.
Nope, that's in her way. That's in their way.
Oh,
Oh, it's okay. I could just put it here.
okay, good.
I
All
love
right,
how
so-
you're so straightforward.
Myra saw that there was problems with the way we did things last time, so she went ahead and, because that's Myra, she does all the stuff for us. So, what she did, this is Streptococcus pyogenes, and so she just drew one line, and so you can see that around the A disc for Streptococcus pyogenes, there's a zone of inhibition, but around the A disc for this other organism,
That's not at all what it looked like what we did.
no. So this is just
Okay.
her, right? So you guys can take a picture of that. Then the other one is Streptococcus pneumoniae, and so the same thing here. So you can see the A disc and the P disc, but here it's got a zone of inhibition around the P disc. Okay?
Which one was it? Oh, the S.
Streptococcus
Put it a
pneumoniae.
little further back.
Okay.
Yeah, right there.
Good.
Thank you.
So you guys, if you put them side by side, you can take a picture of them. And they're pretty good plates. So whatever reason ours didn't work, Myra fixed it because Myra fixes things.
Yeah, we need
So the way we did it was not
Yeah, I'll get
how
them
we
again.
needed it.
I don't know what happened.
Okay.
So it normally works very beautifully.
Okay.
This is Dr. John's method.
Okay.
And so I think Myra did these plates for Dr. John, but when she saw my plate sucked, she said, just use these. Okay. Good.
Thank you.
Anybody have questions?
Excuse me, Kim.
Sorry. Will those be available after you're done talking? Oh, okay.
I'm not gonna, I
No,
mean,
you can't
your
see
plates are over there, but they didn't work all that great.
So we,
So-
yeah,
Oh.
that's to your liking.
You know what you can
Yes.
do in a minute, if you want, just
Oh no, it's on the table
you
though.
can take a sheet of paper like this
Okay.
and then
Yeah,
put
do
them
that.
here.
Okay.
And you can
Write
put
it.
stuff,
Oh,
you
that's
can put
okay.
stuff
Okay,
like
okay,
this,
okay.
and then you'd have
Okay.
them.
All right.
You want me to write it real quick?
Okay.
She wrote the name on
So you can put Streptococcus pneumoniae here, S period pneumonia.
I don't.
Let me do it.
I can do it.
Here, we could just cover it up.
Okay, good. Okay, good.
Smart.
Look at you.
She could be a microbiologist, like he says.
Cut it up because you got your back out.
And like you know.
By the way, that's not my pencil.
Oh my gosh.
5 points.
This is your pencil?
Thank you. That's why I wanted it, but
Extra points.
Well, he stole mine.
Okay, so I'll put them here like this and y'all can come and take pictures of them. Here's your pencil. Okay? All right, now, let's look at these. Then look at the virus group. So remember, this was run in tandem. What are the two tests that are run in tandem?
We can put it over here if you want.
No, because I gotta reach them. Bioescalin 6 point, here, let's just put them here.
Yeah.
That's okay. Bioescalin 6.5, okay? So if we look at these tests together, if you have both of them negative, right? If you have both of them negative,
What does that mean? We'll take pictures.
well, that means it's either group A, B, or C. Okay? Good? If you have one of them positive, bioescalin positive, but 6.5 negative, black precipitates positive for bioescalin,
Hi,
then
Joe.
it's groups D, non-enterococcus, H, or K. Okay? And if you have both of them positive, this is beautifully done. Virus group, you did a great job.
Thank you.
So if you have both of them positive, right, then it is enterococcus, group D, enterococcus. Okay, so these would be like this. This would be almopracticum.
we messed up, didn't we, right?
How did you mess up? These are good.
No. We did.
Okay, so
Six.
if you can look at your own stuff, but these worked really great, and so I'm gonna leave these here as the
No, yeah,
model. This is the model. Okay, good. All right, now let's talk about what we're doing today. Can you put those over there? See how key on. So a couple of things what we're doing today. First of all, we're gonna do antibiotic susceptibility testing. It's also known as Kirby Bauer. Kirby Bauer were the guys who came up with it, the individuals who came up with it. And what we're gonna do is make a lawn of growth, and then
I need it. I'm sorry. Let me take a picture. Yeah.
okay,
Which one is it? She needs a picture.
time. I got time.
It's fine.
Jalen started to take things apart.
I'm
What's
sorry.
called
I thought you meant like
caused a
kinda like
domino
Let me go
effect.
get a picture.
Kind of, because you were
Yeah.
all kind of just bullshit, but
Oh.
thank you.
I'm gonna have to take it, I'm sorry.
Yeah, because Jalen's gonna take it all apart.
I need it.
And Ki Young's not gonna give you a pencil
Oh,
back.
I know, I hate that. And then you're just like, don't know when
Your car,
to.
fine.
I know.
your phone like it was mine.
New car.
Oh, yeah. Yeah. I thought I
Anybody else want to
I
take a
know,
picture
I
before we
think we're
continue?
screensaver.
Yeah.
I guess I might as well for waiting.
This is yours.
But you were like
Well,
holding
let's
over
wait,
my
because
camera.
let me, we're gonna run out of time.
Okay, okay.
Okay, so now Jalen's gonna take her stuff back,
Yeah.
because that's the way
You know,
Jalen.
he is somebody interested in you.
Okay, so let's,
His cousin is,
all right,
mind.
so listen, what we're gonna do is we're gonna do sensitivity testing, right? And so everybody is gonna do one plate. Everybody's gonna be doing, I'll show you how to do it in a minute. So everybody is gonna do one plate. You're either gonna do antibiotics, you're gonna do disinfectants, antiseptics, or you're gonna do essential oils. So I think the Cheskinator brought her own essential oils. There's also some back here. We have orange, we have tea tree, we have eucalyptus, and we have clove.
I love the way that smells.
So you can do either one. So how come this is important? Well, so this was one of my students who I caught in the lab one time crying. I said, what's wrong? She said, my dog has got a urinary tract infection and she's really old. And they're going to charge me $400 to do a culture and sensitivity. And I don't have that money, so my dog is really suffering. I said, just bring me her urine, I'll do it for you. So I put it on a blood plate, and then I isolated the organisms. It was E. coli. And then I did a culture and sensitivity, and so you can see that I put 7 different antibiotics on there. And then I did the measurements and could tell them which of the antibiotics would be effective to treat the dog with, right? And I didn't charge her anything.
But can you prescribe to the?
I cannot prescribe. No, I can't do that. I can do the testing, but I cannot prescribe.
That means she still has to pay. Right?
Well, so that was the problem. The vet called me and said, well, I need to know your credentials. I'm like, no, I have 3 degrees, 3 certifications in microbiology. I do this for a living. She said, well, we don't typically allow outside people to do the diagnostics for you. I said, who does your diagnostics for you? And she said, so-and-so lab. I said, well, I do the same thing they do, just for free. And she got a little grumpy with me.
Right.
I said, look, you can charge them for your evaluation, but you can't charge them for the work because I did the work. So I don't know what she did. But she goes, what's your recommendation? I said, ciprofloxacin.
It's like, you're right.
Well, that's what it is. This is nitrofurantoin here, right? But the biggest, to me, the biggest success was ciprofloxacin, and the dog was older. I didn't want to take a risk. Cipro would take care of a lot of things. I just said, don't let the dog run, right? Because if you give them ciprofloxacin and they run, they can tear their tendons. Happens in adults and humans.
Yeah,
also.
I remember that. I just remember that from college.
So this is why it's important, right? So this is a dog, and I did all of my workup for my dogs, right? So my dogs would get sick, I'd do all their workup, and I would just take it to a vet. Here it is. I just need this, right? And so it helps to understand science. So what we're going to do today is we're going to do this process, but we're not only going to do it for antibiotics. We're going to do it for anything you want to do, and I will show you how to do that in just a second. Okay? So when we make this, what we want to do is we want to be sure that we get the proper technique. So I'm going to show you the proper technique in just a minute. But everything's on the board. You can see that right behind Carolina's ponytail is this little diagram showing that we're going to streak it at 0 degrees, 90 degrees, and 120. And I'll show you how to do that in just a second. And once we get it streaked, then we put the antibiotics, disinfectants, and the essential oils on. Okay? So, we also have, now we're not going to have these because of Riverset, but there's also these dispensing agents. We can dispense them onto the plates. We're going to use the old-fashioned method of just using tweezers to put them on, and I'll show you how to do that in just a second. Okay? But here you can see that this is called a lawn of growth, right? So this is the bacteria that's growing confluently on the plate, and then we put the disc on, and we come back, and we can measure the zone of inhibition. This is a zone of inhibition, right? This constitutes a possible sensitivity to that particular antibiotic. This constitutes a resistance. If the organism is growing right up to the disc, that means that the organism is resistant to that molecule, whatever it is. Okay? Questions? Zone of inhibition. Okay? So this is a McFarland standard. McFarland standards go from 0.5 all the way up to 7. I like to make about a 1 McFarland. Myra is so sweet. You can see that she's already made the McFarlands for us. You can see that these are a little bit lighter in concentration than what we normally would get. Gio, will you hand me one of the McFarland standards? It's over there. No, it's not over there.
What did you say the zone of inhibition?
Yeah, just one of those right there in that green, and that just one of them. Just one of them.
If there is a zone,
It
Oh no,
clears
not that
up
one. Just the one in the little screw top, the black one. Yeah, good. So this is a McFarland standard. This is one that we bought from Rimmel, and you know, if you don't know what you're doing, you can always just compare. This is ammonium persulfate in here. It doesn't change over a period of time. So that's about a 0.5 McFarland, and so I like to target about 1. So Myra makes it about 1. So this is slightly more concentrated. Are you with me? Well, what we're looking at, give that back to him. What we're looking for is so that we can control the population of cells, right? So the turbidity is directly proportional to the amount of cells that are in the population. We don't want to put so much population that the effectiveness of the antimicrobial agent is masked because of the amount of cells that are in there, right? So the antibiotic, the antiseptic disinfectant, the essential oil are going to diffuse radially, right? They're going to go in all directions from the disc. And when they come in contact with the organisms that are sensitive to them, they're going to kill them, right? So there's going to be the zone of inhibition. And we can measure the zone and then decide whether or not that particular whatever is effective against that particular organism. Now we're only using 2 organisms. We're using either E. coli or Staphylococcus aureus, right? And so you can see I've already done this for you once on this presentation. When we went online, I came in and I put all these things together for everybody. And these are your lab manual. This is your lab manual. Right? This is exactly what you should have looked at today. So, using a McFarland, be sure you know what a McFarland is. I don't need you to know it's a 0.5 or a 1, just a McFarland. A McFarland is the standard of turbidity. It's directly proportional to the amount of cells that are in the particular sample. Okay? And so if you think about it, if you have too many cells, then when the antibiotics start to diffuse out radially, they're going to be inhibited by the amount of cells, right? They're going to be used up by the amount of cells. What we want them to do is to fully destroy the organisms where they're susceptible, so we have a nice zone. We can measure that zone and tell whether the antibiotic is effective or not. So our result is going to look something like this. Now this is a much larger plate. We have the standard Petri dishes that have got agar in it, and we're going to probably get about 6 different antibiotics out. Okay, if we want to target that. So here's one of the dispensing. We don't have those, but we're going to do it the old-fashioned way. And then here's a video showing how to measure the zone of inhibition, right? So I have some videos on here you can watch, but I'm going to show you how to do this. And then here are my plates that I did, and so you can see Staph aureus versus E. coli, and you can see these. This is called what? What is this growth called?
Lawn growth?
A lawn, just a lawn.
Oh, lawn.
You can say confluent growth, that's okay. Lawn or confluent, you need to know those terms. And then this clearing is what? What's that called? Zone of inhibition, right? So we'll have to measure those zones in order. Now this is disinfectants, right? And so you can see that they're a little bit more messy because we buy the antibiotics from a company, but we make our own discs for disinfectants and for essential oils. So it's a little bit more messy, and I'll show you how to do that, okay? But here you can see if the organism's growing right up to the disc, that means those things were not good at all against that particular organism. And then here's the essential oils, okay? So you can see here clove looks like it was a little bit better, although it has breakthrough. But if you're using it as essential oil, it's not for medical use anyway. It's more holistically, if you will, okay? So you would measure the zone, and so for this particular antibiotic, you'd measure the zone. So this is a millimeter, so from here to here is 10. From here to here is 10, from here to here is 10. So this is about 21 millimeters in diameter, okay? And so then you would look at a standard. And there's standards for every single group of organisms. So the standards look like this. This is a very, an example of a standard, right? And so here's one for Staphylococcus, right? And then here's one for the Enterobacteriaceae. That's for E. coli. And so the two that you're going to be using today is E. coli, which is an Enterobacteriaceae, and then Staphylococcus aureus, which belongs to this group. Right? So we should be able to measure those zones when we come back on Tuesday. Okay? Any questions? All right. So I'm going to show you how to do that in just a second. Okay? Got some videos for you if you want to watch it. Let's talk about UV.
I don't think so.
So with UV, I've already done the experiment for you and I've documented it. So we're going to do something a little bit different today.
So we're just going to be turning the lights on?
No more.
So here I have 3 different UV instruments that I use. One of them I bought at HEB when they were going out of business, and that's this little handheld one. So why don't we say that the bacteria do this? Keon, inside of the closet there, inside of the room, on the right there's a little bag that's blue. Will you bring that bag out? It's got my wands in it. You see it? Oh no, it's over here, Keon. It's over here.
Over here, yeah. We're still looking for it.
Go, somebody go get him. He's lost. Yeah, he's probably there talking to Myra. So here are my other wands. This is my Mondo wand. I call this the Harry Potter wand. Although it's bigger than Harry Potter, right? This thing will take out some stuff. Okay? So why don't we say you all do this one? Okay?
Do we need like special glasses for it?
Yeah, your normal glasses. You have to wear your normal safety glasses. And then you guys are going to do this wand here. This is the take home.
That's the baby.
Put it in your car stick it to your computer or your USB on your car, and it works, okay? So you guys are gonna do that. I'm gonna show you what we're gonna do, okay? So UV is effective in the UVC range, right? And so if we think about this, UV affects microorganisms because it interferes with the replication process of DNA. It causes thymine dimerization, right? And so this is the one we want to target, UVC. That's the one that's most effective against the organisms. Okay, it causes a thymine dimer, so you can see that it binds up these thymines right next to each other, therefore the DNA cannot be replicated and the cell dies. It's very effective. However, in order for UV to be effective, it has to be able to reach the cell. And so I always tell people that UV is only a disinfectant tool. It can never be a sterilization tool, because there's no way that we can guarantee that it's going to hit every single cell. Okay? Because again, it's flowing, the light pattern is flowing wherever that light hits, it's going to do its job. But if it doesn't catch a cell, it's not going to kill that cell. Okay? And you'll see what I mean in just a minute. So we want to target about 200 to 280 nanometers for the lethality of the UV. In this lab, we use 265. I always try to buy UV lights that are 265. To me, I have this big old thick book that says germicidal agents and how to use them. And so in there it says the most effective UV wavelength is 265, and that's what I've always used. Okay? So I think I told you that my wife, when COVID hit, she said, "Are you gonna?" And I was like, I got tired of using bleach every time, so I bought the Mondo One. I bought that thing, and anything that came in, you know, it was. I would just go over it about a minute, kill everything.
I hope I knew it.
Well, only until she was satisfied, because I said fomites don't play a role, and she said, "Really? You're gonna talk theoretical with me about this? I'm trying to protect us as a family." I'm like, okay, I'll do it. So I did it. But now I don't have to, because now the papers are written, and I'm like, I put a paper on her desk and I'll run away and she'll read it. "Did you read this paper?" I'm like, "Yeah." So I was right? But anyway. So we're going to expose this. And so here's a video exposing bacteria, one half of the plate to UV, the other half not. And so you can see how it would grow. What I did was I did this particular little experiment. It's really an exercise because I know what the result was going to be. But I did this when we shut down. I came in for 2 weeks and I built all my online stuff. I put it in the lab manual, and that's what it is, right? So you can see that I did a control so that it wasn't at all exposed to UV. I did in 1 minute, 3 minutes, 5 minutes. I did 10 minutes without the lid, and I did 10 minutes with the lid, because plastic and glass stop UV, right? And so I wanted to show you how that works. So here's my results, right? And so let's talk about these results, because you're going to be responsible for these results. So if we look at these, you can see E. coli's here, and this is Bacillus subtilis. And so these plates represent control. These plates represent 1 minute. These plates represent 3 minutes. These plates represent 5 minutes. These plates represent 10 minutes without the lid, and these plates represent 10 minutes with the lid. So if it's got the lid on it, the lid is protecting it against UV. UV doesn't penetrate the plastic, and so the organisms grew just as well as the control did. So that just means that I had that's a Lana growth that I made. And so you can see that this is going to be, this particular little experiment got positive controls, it's got negative, it's got everything. So it's working. So now we just need to look at the data. So first of all, why did Bacillus subtilis grow so much more than E. coli? You can see here at E. coli, there's no growth except on the edges. Now this thing is exogenous. It's something that was in the air, it fell on the plate. This is at 1 minute. You can see it's only growing around the edges. Here is a couple of colonies here that are exogenous. They're not E. coli. Here is E. coli growing around the plate, around the edges. And then you can see here is a little bit, maybe right here, of edge growth. But if you look at Bacillus subtilis, they just grew crazily, right? Even at 10 minutes, a lot of the cells survived. Why is that?
What's in the agar?
The agar is just nutrient agar, It doesn't really have any kind of advantage to anything. But what has an advantage is what Bacillus produces. What does Bacillus produce that E. coli doesn't? Starts with an E, ends with an E.
Enzymes.
Huh?
Enzymes.
No. Has an N in it. Endospore. That's right. So endospores are produced by Bacillus and Clostridium, and therefore the endospores are refractive, they're protective. UV didn't destroy them. But UV destroyed everything in E. coli except the ones on the edges. Can you give me a hypothesis about why there is growth around the edges of the plates?
It was harder for the light to reach the.
The lights couldn't reach it, so here's the plate, and when the light came down, the edges produced a shadow, and the cells that were right next to that were protected because UV didn't touch them, because that plastic edge produced a shadow and it protected those cells. That's good. Good work. Okay?
So if you just got a light that was more condensed and kind of pointed it around.
Yeah, you would do that, except nobody does that. Right? So yeah. Okay? Questions? So again, we've covered all these things, right? And so here's a guy who basically says we should be using UV for lots of different things. Instead of antibiotics, we should be using UV.
I was thinking that, but like.
But not like the Trump wants to do it, not that way, but not internal, but on the outside. Okay? Good?
You internally.
But wouldn't you kill the good bacteria too, though?
What's that?
Wouldn't you kill the good bacteria too, though, on the
Mm.
your microbiome?
Yeah. You can watch him. It won't be on the practical, but if you want to watch him, he's pretty cool to think about. He's got really great ideas. Okay? So anybody have any questions? So here's a citation for my big book of disinfectants and how to use them, right? Here it is, but and there are the YouTube channels that I used up. And so let's actually do the lab, and I'm gonna do it right there where Keon is. Let me get suited up real quick. Did it break? It's good over here. What is that? Oh, the Chescanator stuff. That smells really good. That makes me hungry.
Good. I smell.
I'm on Pepper's chicken.
Yeah, exactly. It's a common spice in Asian cooking.
Lemongrass?
Yeah.
Yep.
Kiss my ass. Give it back. Yeah. Give it back. Give it back.
Are you afraid for her?
Me too. I went to bed at 2 in the morning.
Is it even right, Cheska?
That's when I finished my questions. I saw Cindy typing and we were finishing together.
I don't have my phone on me. You have your phone?
What?
Over here, there's
You're 59 actually.
Okay, that's right.
Cold, I'm tired. I want coffee. I'm starving.
It's a little after.
I feel you. Oh wait, so he's going over there after all? Or- Are you for real? Because I thought so.
He is. He just,
He just said he was gonna do it over here.
I don't know, 'cause I can't get it.
She said
That was a date. Oh, yeah. Hey, give me the hat. That's brutal.
This is mine.
Oh, I'm so sorry.
I don't want Gio to feel like he's left out. I never do anything over here.
You probably called.
I thought you were gonna take your spot. I was like,
All right, everybody come on over here. So what I'm going to do is I'm going to make all the plates for you all. Carolina, pick an organism. Okay. Good choice. So she says E. coli. So I'm going to label these E. coli. So I'm going to do antibiotics. You guys target something else. You guys either do the essential oils or you do the disinfectants, okay?
Okay.
So this is going to be antibiotics and today's date. And today's date's the 23rd. And then this is UV. and this is the large wand. This is the control. This is UV. This is the large wand, and this is 30 seconds. And then this is UV, the large wand, and some minute. Okay? So, if you don't have to turn the fire on for this work. You'll only have to turn the fire on to place the disc on the plate. Okay? So I'm going to use a sterile applicator stick. And what I'm going to do is I've already got my McFarland for E. coli. That's the one that Carolina has asked for. What I'm going to do is I'm going to streak each of these plates at 0, 90, and 120 so that I have a really nice confluent growth or a Lana growth, right? Sterile, I'm going to go in here. I'm going to get it wetted really well. Wetted is a term that means that you're going to get it completely saturated. It's so saturated, I'm going to go ahead and wring out some of it up top. And then I'm going to take the plate so you all can watch me do this. And I'm going to take the plate, and what I do with this is I draw one line down the middle. And then I will streak it. This is at 0.
For a lawn.
And then this, I'll turn it. This is at 90.
You don't want it again?
Don't have to.
You
So
don't
this
want
is
too
at
much
90.
organism, right?
Huh?
You don't want too much organism, right?
Right. And then I'll turn it 30, and this is at 120. And so once I've done this, good technique means that I just kind of wring the edges so that I get off the extra moisture so it's not going to run around. Okay? But then I'm going to do this
He's gonna do the end.
3 additional times.
Alright.
You're dipping the same one in there?
Same one, because I'm using the same organism.
Oh, okay.
Right? If I was doing different organisms, I'd
Right,
get a
got
different.
it.
But again, making one line, streaking at
Yeah.
0, at 90. And at 120.
Which one did you just do?
Which was the first one you did?
What do you mean?
The plate.
So these are I did the antibiotics first, now I'm doing the UV ones. Okay, so again, I've got 2 more plates to do. So one line down the middle, then I streak it at 0. At 90. And then at 120. Hey, Amira, will you bring out one container of just disc, because we have some people who brought their own essential oils? Thank you. I have one more plate to do. So again, 0.
So why did you want to use a sterile, what's it called, instead of an inoculated loop?
Because I want to get a really nice big flat surface to spread the organism over the entire plate.
Okay.
Okay?
And a loop would
With the
kind of
loop,
add
if you
to
do it with only the tippy top, you're only going to get that. And so it's not, the loop is more for isolation. Here I want a confluent growth.
Okay.
That's a beautiful question. That's going to make it on the practical.
Great.
She's your buddy.
I know, right?
So this is at 90 and then at 120.
Hey, we
Thank you,
gave
Jake.
people 2 points, right?
We did
Okay.
it.
One for you.
So once you do that, this just goes in here. Okay? We'd like to go ahead and recycle these. Okay? All right, now I'm done. So these 3 plates, this is the control for UV, this is at 30 seconds, this is 1 minute, is going to be given to the Prion group, and they're going to expose that light. Now, when you use those UV lights, you want to be sure that you're wearing your covering for your eyes, because those UVs are powerful and they will destroy your retina. If you are afraid to do it, let me do it for you.
That's why I was asking, like, why aren't we gonna use like at least like shaded glasses instead
You're
of?
supposed to have shaded glasses. You're supposed to have side shields.
But that
You mean like not clear, right?
Glass
Like
or
our goggles
glass or
are clear.
plastic stops
Oh, because
UV.
yeah, that's right. Okay. That makes sense.
This works.
Yeah.
Okay? All right, now, For the antibiotic one, Gio, pick 6 antibiotics for me.
6?
6.
No, he hasn't done it yet.
Oh, you got what?
Oh no, that just went really high.
Okay. So, let me check. Gio has selected oxacillin, Cipro, nice. Ceftal, nice. Some good ones, buddy.
Thanks.
Levofloxacin. You have 2 fluoroquinolones. You want to pick another?
Yeah.
Okay. Ooh, nitrofurantoin. Nice. You have 2 third-generation cephalosporins. Give me another. That's a third-generation cephalosporin.
Yeah.
I mean, even though they're like second and third generation, wouldn't they have like a different effect on it?
Probably not.
Probably not.
Good question?
Ticarcillin. You're going big gun on me, buddy.
Yeah.
Dang. You're worried about this one, aren't you? That's the way one of the docs that I used to work with. Oh, you're bringing out the big guns. I'm like, yeah. I like the patient. Okay. So the way you're going to do this is you want to put these discs on this plate equidistant. So again, you have a lawn of growth that I've already put on here. Now you want to put these discs on there equidistant. And so what I like to do is I like to get the pointiest thing I can find. That's about it. There's one I got. Okay. Get it in the alcohol, hit it there, let it burn off a little bit. That's a really good job for a pyromaniac. Lakeisha, I know you well.
I was like,
So ticarcillin, take a little bit, stick it off one end, pull it out like this. And then find a nice place for it on the plate. Stick it on there. And once you stick it on there, just push it just a little bit. Somebody didn't push them last time, they all fell off. You want to push them so that they make contact and they're not going to fall off. Okay? So that's one. Next one is second generation Seth. So I'm going to put this equidistant, right, somewhere. Put it down just like that. This is nitrofurantoin. I love the way it looks. It always has this rust-colored appearance on there. One of my favorite drugs.
which one's your favorite drug?
Nitrofurantoin, Macrodent. See how I'm putting these down?
Okay.
Oh, arm's broken.
So how does the heat not affect the antibiotics?
It doesn't. It doesn't get hot, Jalen. It just burns off the edges. So in case there's any of the molecule on there, it just gets rid of the molecule so there's not any kind of cross-contamination.
Okay.
And I'm not touching the plate, so I'm not getting any bug on it.
Same as the stuntmen, they do fire on their body, but they took out for a couple minutes.
I don't know anything about stuntmen. I got to tell you, I don't know anything about stuntmen. What's going on here? Ciprofloxacin. And then the last one. Sulfa.
What is it?
A sulfa. Uh-huh.
So.
Oh, it's oxacillin. Okay. Whatever you do, if there's a flame on this particular metal, do not put it in the alcohol.
Oh.
Okay?
Please don't.
Is DLC the flammability factor?
Or your glasses won't protect you.
That's right. There will be a big kaboom. Okay? So you can see how I've arranged on this plate. You guys will do things like that. Okay? So, do you all want me to do these for you so everybody can see how I do them? Okay. So let me go ahead and do let me go ahead and do the UV for you all. Oop.
Should we be wearing our glasses?
Yes, everybody put your glasses on. Okay. So everybody got glasses on?
Yeah, that's what I was saying.
Alright.
This be our last day wearing glasses.
What's that? This is our last day to wear glasses. Yep. Okay. This is the control. It doesn't get exposed to UV. I'm going to put it over here on this tile right here. Okay. This one's at 30 seconds. This one's at 1 minute. Who can time me? Can anybody time me?
I can.
Okay. Let me know when you're ready.
Okay, where is it? For how long? 2 minutes, you said?
1 minute.
1 minute?
Tell me when you're ready.
Okay, on your mark, get set, go.
So this is all you do. And so you leave it here for a minute. Now, if it was Jay Lynn, she would be moving it around all over the place, right?
Jessica should be.
Oh, I didn't mean to laugh. I didn't mean to laugh. That's my fault.
I mean, is it important that we just not move it around? Does it
No,
matter?
you can move it around if you want.
Okay.
The reason I don't want to move it around is because if I don't hit every single spot, I want to prove to you all that it's not a sterilization method, right? At 1 minute, everything on here should be destroyed if
So-
this UV is effective.
How possible is it that you can actually destroy everything, on everything that you're trying to destroy?
Like, what's the percentage?
Flat surface, 99.9%.
But surface like that though,
Less than that.
That's over.
Okay, tell me 30 seconds now. Let me know when you're
That's on
ready.
the Albert plate on there. It wasn't right. It didn't have like
It's because it was causing the shadow?
the yeah, something like that. Yeah.
When you're ready.
Alright, on your mark, get set, go.
I know it's gonna
Do you know what?
Okay, it's over.
Okay. So I've done everything for you guys. All you have to do is one plate and do your antibiotics or whatever. Okay? Everything else is done for y'all. So the viruses you guys are using the small one, and then in the middle here, the bacteria are going to use that as seen on TV one. Okay? Now I'm going to take my batteries out of my presenter, so that you guys will put those batteries in there and it'll work.
In
Okay.
ours? In the
Huh?
virus or the...
For the bacteria.
Oh, okay.
Okay? Everybody good? All right.